Okay, so now we're going to be having a look at running the same search that we ran before at NCBI at the EBI. So I'm on the EBI homepage, and so at the top of that homepage you'll find that there's a search bar. And what we're going to do is we're just going to put in FoxP2, and we're going to search across all the database. So this is again the portal that allows you to search across all the databases and resources at the EBI. Okay. So this is the results page that we get back. So it's kind of similar if you think about NCBI. What they've done is on the left-hand side, it tells you the number of hits that have been returned from the various different larger types of resources or categories of resources at the EBI. And then on the right-hand side, it sort of breaks down all those entries a bit further. So you'll see that the first section is gene and protein summaries. And the first hit here is forkhead box 2, and it's FoxP2 in human. Okay. So before we have a look at that, I just want to show you a bit of in more detail of the different results. So essentially, again, remember the portal allows you to search across all the resources at one time. So in total, there's over 2,800 hits that we've got back using FoxP2, and again, these will return relevant results in the different categories. So we have 71 hits in genomes, we have 1,200 hits in protein sequences, we have 388 hits in literature, etc. So again, remember we searched for FoxP2, we didn't limit it to human, so all these entries in here would be related to FoxP2 across different organisms. So what we're going to do is we're going to click on, since we again interested in the gene, we're going to click on forkhead box P2 under the gene entry. Okay. And this is the result that we get back. So it's slightly different to NCBI. So again, we have these different sections, which basically show you the different information that is known for FoxP2. So the first section is gene information and sequence. So this tells you straight away a bit about FoxP2. So it says FoxP2 spans, this is the length, and it's on chromosome, found on chromosome 7, and it gives you the actual position of the gene in terms of the genomic sequence. It then says it has 26 transcripts. So remember I said there was some evidence from NCBI for alternative splicing, and it contains 98 exons on the forward strand. An important thing here is it says annotation for this gene includes both automatic annotation from Ensemble and Havana manual curation. So remember at the beginning of the session I spoke about curation, and that some resources are curated and some resources are non-curated. So in the case of this entry for FoxP2, or the annotation information for FoxP2, the annotation information or all the information for FoxP2 has actually been manually curated. So Havana is a project that has set up to manually curate all genes in humans and also across mouse and some other model organisms. So this is an ongoing process whereby we actually have human beings going through data, ensuring that all the data is correct and that the annotation and sequence data for all human genes is very accurate. Okay, and in this case, the Havana project aims to do that for all human genes, and they're in the process of completing that. So you'll find that for most human genes, when you're searching for information, that there will be a Havana manual curation of that particular gene. Okay. Then we have two links here. So the one is to view the gene sequence in Ensemble. So Ensemble, I'll speak about that a bit later, and we'll be doing it. I think I mentioned it before, but we're looking at Ensemble in detail in the genomics module. But essentially, it is a way of visualizing where the gene is and all the information for a particular gene in a genomic context. So we can view the gene sequence in Ensemble. We can review the chromosome region for the gene in Ensemble. Okay. We also then have a variation section. So this tells us a bit of an overview. So it says FoxP2 has 27,589 SNPs. So those are single nucleotide polymorphisms. And again, if we click on the link, we can then view those polymorphisms and more information about the genotypes as well as any disease association in Ensemble. So what you're seeing here now is that EBI uses Ensemble for viewing a lot of the information for any gene entry. So any gene entry, you can view additional information on that particular gene entry using Ensemble. We then have an ortholog section, so it tells you are there any orthologs in other organisms, FOXP2, and again we can view this in more detail in Ensemble. It also identifies paralogs, so these are sort of regions that, in paralogs that have been identified in humans for FOXP2, and then there's a regulation section. So this is something that EBI has worked on quite extensively, is they've tried to identify possible regulatory elements that are involved in regulating the expression of FOXP2. And again, we can click on that and view more details about that in Ensemble. So like I said, we're going to have a look at Ensemble in detail in the genomics module. So I'm not going to go into detail now, but I just want to give you an idea of what it looks like. So this is the example of if we link out to Ensemble and have a look at the gene entry page for FOXP2. Okay, so it's very similar here. It gives you the gene name, it tells you the description, synonyms we've seen before, it tells you where is it located, it's on chromosome 7, it tells you a bit about the different transcripts, paralogs, orthologs, etc. And in this case, what we're looking at is some of the alternatively spliced transcripts and some information about the length of those transcripts, how long is the protein that it encodes, etc. So again, we're going to come back to Ensemble and figure out, and we're going to do quite an extensive practical on how to access and view information in Ensemble, but just keep in mind that EBI relies heavily on Ensemble for viewing a lot of the information for a particular gene. Okay. So if we go back to, so the information I was showing you on this page allows you then to access further information on FOXP2. So this is the entry page for FOXP2, and if we're interested, for example, in looking at expression, we can click on the expression tab and it will give us information about what is known about where this particular gene is expressed in the body, and what function it's involved in. You can also click on protein and it will tell you a bit about what is known about the protein, where it's localized, where it's produced, and then what diseases are associated with the protein. Okay, we then also have information on protein structure. So if the structure of FOXP2 has been solved, and in this case it has been solved, in this case it's bound to a particular bound to DNA. The structure has been solved bound to DNA, and we could actually have a look at more information on the structure for FOXP2. And again, like NCBI, we also have associated literature that has been used, studies that have generated data that's been used to annotate the FOXP2 gene. Okay, so these are some of the other popular resources at EBI. Again, I spoke about Ensemble, so I'm not going to go over that again, but we'll be having a look at it in more detail. There's also Uniprot, which is the universal protein resource for protein sequence and functional annotation data. And again, we will be having a look at that in the last session of this module. There's PDBE, which is the collection of 3D structural data. And there's Interpro, which is the database of protein families, domains, and conserved sites. And again, we'll be having a look at that in the last section of this module. And then finally, I just want to speak a bit about specialized databases. So remember I said before that there are these more generalized databases and there are specialized databases. And a lot of these databases are housed at GenBank and EMBL. Okay? And some of them are housed independently at different sites. So most of the sequences or sequence data is housed at GenBank or EBI. And these may contain whole genomes, but they are specialized resources. And one of the advantages of identifying these specialized resources, let's say, for example, if you're working on HIV, rather than going to GenBank and trying to access sequence data and annotation for HIV, you'd rather go to try and find a specialized database in order for it to be easier for you to access information quickly and easily, rather than having to sift through large databases that contain information and sequences that you're not interested in actually using. So these are just a few examples of some of these specialized databases. So there's a Plasmodium database. There's a whole collection of specialized databases housed at the Sanger Institute. There's a hepatitis database. There's an influenza database. There's an HIV database at Los Alamos. So essentially what I'm saying is that if you are dealing in a specialized area, have a look and see if there is a specialized database that will allow you to more easily access the information that you're looking for. And also, what these specialized databases have done is that develop specialized tools to do the analysis that is unique to analyzing the data in that database. So I'd encourage you to have a look for these specialized databases. And again, you can go back to the nucleic acids research database for issue to try and look for these specialized databases. So I think that's all I want to talk about in this lecture. And then finally, just to summarize before we end. So hopefully I've been able to show you that it's very important to understand what data is out there. There is a large amount of data out there, and it's very important to first know what data you're looking for, and then select the correct or the easiest resource that's out there to be able to access that data. I've hopefully shown you that primary databases store raw sequence data, and then we have a number of secondary databases that provide information and annotation of the sequence data. It's important to know how and where data is stored, and I've hopefully convinced you that the NCBI and EBI basically contain most of the information that you'll ever need to access for most bioinformatics projects. And I think by playing around with it, playing around with these two resources is what we'll do in the practical session. And hopefully by the end of that, you'll find your database or your resource that you find most familiar and easy to use. So what we'll be doing in the practical is having a look at these two resources and extracting data from them and becoming familiar with how to extract data from these two different resources. Thank you.